In this video, I'd like to talk about how to express some protein of interest and also transgenics, which we'll learn are closely related. So what I mean by expressing a protein of interest is, let's say we have some gene that we want to study and we want to get a bunch of the protein that it encodes. So we need to somehow cause some organism to express this protein. Okay, so we already, as we've learned, so the first step, it turns out, is we're going to use PCR to get a ton of copies of it, and then we'll figure out how to express it. So in the simplest case, so we can use this thing called a plasmid, and plasmid basically just means a circular DNA. And so here, so a circular DNA can have sort of anything on it, and so we're going to, in this case, if we have some gene we want to express, we're going to just have a circular plasmid. It doesn't really have much of interest, except for that it has a promoter. And so then what we want to do is add in DNA for our gene of interest so that it fits in next to the promoter. So what this is going to mean now is that this promoter is going to cause transcription to start here. And now because our gene is right next to the promoter, our gene will get expressed. And eventually, so it'll get transcribed and then translated. And so then we can take this plasmid with our gene in it, and we can introduce it into bacteria, and voila, we have bacteria expressing our gene. But now we need to figure out sort of how we do this. Okay, so how do we get our gene into the plasmid? So here we're going to learn about sticky ends, we're going to learn about restriction enzymes, and we're going to learn about how we can use the plasmid in the gene to make a construct. Okay, so the issue here is that we want to be able to take these two pieces of DNA. So we need to take this piece of DNA and this piece of DNA, and we can need to somehow insert this one into that one. So we need to kind of cut this one open and then stick this one in, and then close up the breaks. So the way that we're going to do this is we're going to use double strand breaks with sticky ends. So what I mean here is, if we imagine that we have a piece of DNA that's double-stranded like this, if we break it here and here, then what that means is that's going to give us two pieces of DNA which have these little overhangs, so we call these sticky ends. And if we think about it, if we have two pieces of DNA, this one and that one, so we said that we can break it, but then if we have a break, then it's going to want to get back together because this AATT is complementary to this AATT. And so we can also use DNA ligase if we have these breaks to repair these breaks and make this into a full strand of DNA. All right, so we're going to learn in a second how to make these breaks, but once we have these breaks, then we can allow them to repair, and then we can use DNA ligase to fully repair them. Okay, so restriction enzymes are these enzymes that cause sticky end breaks at specific sequences. So why do these things exist, right? So we're going to talk about how we can use them, but why did they exist initially? We think that the reason that these exist is that there's ongoing war or competition between bacteria. And so we think that bacteria use these as a tool, as a weapon to destroy unrelated bacteria. They're going to cut up that bacteria's DNA and destroy the genome, and therefore not have to compete with that bacteria because they've killed it, or maybe even eat the bacteria. So the strategy is that we've got an enzyme that cuts specific sequences, and then over time, the bacteria that encodes this enzyme, that has the gene for this enzyme, will have evolved few or no copies of the specific sequence. So that would mean a given restriction enzyme will cut at a given sequence. So here's a bunch of different restriction enzymes. And then they all cut, you can see they cut at different sequences. And so what we have here in the table, we have a microorganism, and then the restriction enzyme that was isolated from that microorganism, its preferred cutting site, and then the cleavage products. If you look at it, this and that, you can work one out for the other. If you cut here and you cut here, then you end up with these two products. Okay, so then what we're going to do, basically, is if we want to contain, so here it says DNA from species 1 and DNA species 2, but this would be the sort of any two DNAs that we want to combine. So this might be the plasmid, and this might be the DNA that we've PCR'd up. And so what we can do if these two DNAs have the same cleavage site, if they have the same site, if they have the same 6 nucleotide site, and if we have a restriction enzyme that we know cuts that site, then we can do the following. Okay, so we're going to take our two DNAs, we're going to digest them both. Okay, so they're going to digest both so that we end up with these sticky ends. Okay, and then if we mix the two of these up, well, sometimes the blue is going to come back and re-anneal to the blue. Sometimes the orange will re-anneal to the orange. But sometimes we'll get these hybrids where we get one DNA joining to the other DNA. And so then we can re-anneal this, and we end up with the DNA from one species attached to the DNA from the other species. And moreover, if we think about that, so if we cut a molecule on both ends, then we could actually get it inserted. So let's think about that for a minute. So let's look at how that works. So if we have a plasmid here, so if we can cut it with EcoRI, one of these restriction enzymes, and then if we cut our DNA to be inserted also with these enzymes, then we end up with these matching sticky ends. And so sometimes what will happen then is that this pink sequence will get inserted in there, and we'll end up with this recombinant DNA. And then with ligase, then it's a permanent double-stranded DNA, and we have now taken our gene of interest and introduced it into our plasmid. Okay, so then basically the short story of how we're going to put our gene in the plasmid is we're going to use this trick with restriction enzymes, and so now we can go with our plasmid and our gene and our promoter. So we've got our first step, we've put our gene into a plasmid. And then we said we can just take the plasmid and put it in bacteria. So we've got one more problem here, though, is that if we cause our bacteria to uptake our plasmid, not all our cells are going to end up with the plasmid. So we'll end up with some that have it, 1, 2, and we'll end up with some that don't have it, 1, 2, 3, 4, 5, 6. So then what we need to do is we need to find some way to select the ones that have our plasmid. And to do that, we're going to use a somewhat more complex plasmid, so a plasmid that comes with an antibiotic resistance gene. So this is a gene that encodes a protein that makes a cell resistant to an antibiotic. Okay, so now we can do the same thing. We start with this plasmid that comes, it ships to us with a plasmid, and then we add our gene in, and now we have a plasmid with 2 genes, 2 promoters, expressing our gene of interest and our antibiotic resistance gene. Okay, so now we have the same thing. We get our bacteria to uptake the plasmid, but only some of them do. But now what happens when we add antibiotic? Well, when we add antibiotic, this bacteria is going to be killed off, this bacterium is going to be killed off, this bacterium is going to be killed off. But we said that this one is resistant and this one is resistant. And so now we end up with a culture that's fairly pure of bacteria with our plasmid that contain our plasmid. So this way we get rid of the bacteria that do not contain our plasmid and end up with bacteria, only bacteria that do. Okay, so that's how we can take a gene and introduce it into a bacterium. And this is very powerful because we can grow up E. coli, the model bacterium E. coli, at really huge densities and huge numbers. And so we can get it to express a lot of protein for us if our main goal is to simply isolate that protein. But it's not quite as simple for like humans, plants, and other eukaryotes. And the reason is that we can't really use plasmids. So as we'll learn when we study mitosis in these organisms, in eukaryotes, linear chromosomes have to line up when we do mitosis, when we have cell division. And so if we have this kind of a plasmid, this round circular this circular DNA, that will eventually get lost. So that's not going to be enough. So we need some way to actually get our DNA into the genome, to actually get it into one of these eukaryotic chromosomes. And so we already said that in this case that we can steal, and we didn't talk about it, but we steal antibiotics, which are products that evolved in microbes to kill other microbes. We stole the idea of restriction enzymes, which are enzymes that evolved in microbes to kill other microbes. And now we're going to steal another weapon. So this is a weapon that is a common strategy of plant parasites. So this is a way, so for organisms that parasitize plants, they're going to feed on or take shelter in the plant. And so what they do is they induce the plant to activate various hormones that lead to growth. So these here, we're looking at what these are called galls. And these are basically tissue growths, sort of like tumors, that are then exploited. So insects or nematodes may actually gestate their larvae in here, or bacteria may grow in here. And so basically the idea is to stimulate the plant to direct resources, to direct growth here that will nourish the parasite. And so the question then is how do they do it? So actually there's active research going on at our institution. Ed Connor's work here at San Francisco State is trying to figure out basically how did insects figure out how to do this trick. But we know a little bit about how the bacterium, Agrobacterium tumefaciens, how they do it. So what they do is they infect wounded plants, and they transfer a plasmid DNA and this protein into plant cells. So the way that this works is we've got a plant, and then the bacterium is here. The bacterium actually transfers this plasmid in this tDNA into the cell. And then this tDNA contains the information necessary. It has a mechanism to actually integrate into the chromosome. You'll see that the red sequence is now here in the chromosome. And so what this means now is that the cell will actually be expressing some of the genes that has been transfected into the cell. So what we have here is a natural strategy for a bacterium to transform the genome of a eukaryote, which is exactly what we want. And so now, geneticists, we've hijacked this mechanism. We've stolen it as a way to transform plants. And so here we have a somewhat more complicated plasmid. But let me just highlight some things. So we've got our gene of interest, and then the promoter before it, and the terminator after it to terminate transcription. This is just the origin of replication to make sure that this sequence will get replicated. And then we have some various other things. But let me point out the bacteria selectable marker. So this is a resistance gene for one antibiotic to make sure that the gene is in our bacteria. And then we have a second resistance gene to make sure that the marker is in our plant or is in our eukaryotic cell. Okay, so let's just go through this step by step. So we're going to do our PCR on our gene of interest. So we're going to get some DNA of interest with a huge number of copies. So then we're going to add that to our plasmid, and we're going to make a plasmid with our gene of interest. Now we're going to take that plasmid with the bacterium, and now we'll have our bacterium carrying that plasmid. And we can purify it and get rid of the bacteria that do not carry the plasmid by selecting with one antibiotic gene. One antibiotic. Right, so only the ones that have the plasmid that carry the antibiotic resistance gene will survive. So then we're going to grow up the bacterium. So now we end up with lots of bacteria, with lots of plasmids, with lots of our gene of interest. Now we're going to add those plasmids to the plant. We're going to incubate embryos or seeds of the plant. And then we end up with a plant with our gene of interest. But of course, this doesn't happen to all plants in our treatment. And so we can select the plants that have our gene of interest with antibiotic 2. And lo and behold, we have a genetically modified eukaryote, a genetically modified plant.